Hi guys, and welcome to our module on the kidney and the ureter. So in this module, we're talking about everything there is to know about the kidneys and the ureter. We'll talk about the gross anatomy, the blood supply, nerve supply, lymphatic drainage, the histology and the embryology, and then we'll end the session by talking about some important clinical significance. The kidneys and the ureter are part of the upper urinary tract. The urinary tract tends to be divided into two parts, the upper urinary tract and the lower urinary tract. The upper comprises of the kidney and ureter, which we'll be focusing on in this particular module, and the lower urinary tract comprises of the bladder and the ureter, which we will talk about in the fourth module under the aspect of the pelvis and perineum. The kidneys and the ureter, just like the rest of the urinary tract, would be involved in the production and transport of urine. So urine is produced in the kidney and is transported from the through the ureter from the kidneys to the bladder for storage. So let's focus now on the gross anatomy of the kidneys. The kidneys are bilateral retroperitoneal organs found in the abdomen. This means that there are two kidneys, right and left kidneys, and kidneys that are lined anteriorly by the peritoneum but lacks a peritoneal lining posteriorly. Hence the reason why we call them retroperitoneal. In terms of dimensions, the kidneys tend to be measured with a length of about 10 centimeters, a breadth of width of about 5 centimeters, and a thickness of about 2 inches. So the kidneys functions as an excretory organ in the sense that it helps in the removal of excessive waste, such as water, salt, and then proteinous substances or waste of proteinous metabolism from the blood. So this is done through the process of ultrafiltration at the glomerulus and renal secretion at the tubule. So as it's doing this major form of excretion, it is also returning nutrients and chemicals back into the blood through the process of reabsorption. So just note that the kidneys are particularly involved in the formation of waste, and this waste comes in the form of urine. In terms of location, kidneys are located in the lumbar regions. So we have the right kidney located in the right lumbar region and the left kidney located in the left lumbar region. They are found largely in on the posterior abdominal wall. They lie on the posterior abdominal wall within the abdomen. So unlike other organs like the intestine, like the large intestine, the ascending colon and the descending colon, those ones are found very closer to the anterior abdominal wall. The kidneys, whereas, are found close to the posterior abdominal wall. They extend from the 12th thoracic vertebrae down to the third lumbar vertebrae. So if I'm to outline the different levels at which the kidneys tend to be located, we have the highest level being the 12th thoracic vertebrae, and then the lowest level being the third lumbar vertebrae. Structurally, the kidneys are reddish brown ovoid shaped organs that have very unique features externally and internally. We'll talk about each of these structural features. We'll talk about the external features of the kidney first, and then we'll talk about the internal features of the kidney, which is shown in the image below. So externally, I like to talk about the kidney in terms of its four major aspects. We talk about the surfaces of the kidney, the poles of the kidney, the borders of the kidney, and then the layers of the kidney. We have two surfaces of the kidney, the and also two poles, two borders, and then finally four layers. We'll talk about each of them one by one. The two surfaces of the kidney includes the anterior and the posterior surface. Why this is important is because of the structures that tend to be related to the kidneys. So we have structures related to the anterior surface of the kidneys, and then we also have structures related to the posterior surface of the kidneys. We'll talk about that when we get to the aspect of relation in this model. Then the kidneys also have poles. Yeah, two poles for both of the kidneys, the superior pole, which is the pole that is pointing upwards. Um, then we have an inferior pole, the one pointing downwards. In terms of the superior pole tends to lie at the level of T12 in the left kidney. So if it's the left kidney, which is a little bit higher, the superior pole is going to be found at the level of the 12th thoracic vertebrae. Whereas on the right side, the superior pole of the right kidney is found at the level of the first lumbar vertebrae at L1. The inferior poles, the right and left inferior poles tend to also differ in terms of the vertebral level. For the right kidney, the inferior pole or lower pole tend to be located at the third lumbar vertebrae at L3, and then for the left kidney, the inferior pole tend to be located at the level of the second lumbar vertebrae, which is L2. Let's talk about the borders of the kidney. The kidney has two major borders, a medial border, which is concave in shape. So if I'm to draw an outline kidney, it has this bean-shaped appearance. So the bean-shaped appearance will make it have a medial and a lateral border. So the medial border is this indentation, this concave indentation of the kidney. So it's concave, and the reason why there's this indentation is because there's a space that is formed by this indentation. The space that is formed by the indentation on the medial border is known as the renal sinus, and it serves as the entry point for various structures into the kidney or structures that are coming out of the kidney. The the medial border also contains a vertical cleft, also known as the renal hilum. For the lateral border is convex in shape, and it is covered by numerous tissues that tends to form what we call the layers of the kidney. So surrounding the lateral border are layers of tissues that we call layers of the kidney. We'll talk about that in the next slide. One other thing to note is that the medial border tend to lie at certain vertebral levels, depending on whether we're talking about the right kidney or the left kidney. So if it's the right kidney, the medial border of the right kidney lies at the transpyloric plane, or at the junction between L1 and L2, whereas that's the left kidney, whereas the right kidney tends to have its medial border lying at the second lumbar vertebrae, the level of the second lumbar vertebrae, or L2. So let's talk about the layers of the kidney. Remember I said that the lateral border of the kidney is surrounded by multiple layers of tissue. So what are these layers of tissue? So the layers of the kidney include four major structures that tend to surround the kidneys from inside out. So this includes, first of all, the renal capsule. That's the first layer. So the renal capsule usually tend to be a structure that is regarded as part of the kidney, but in truth is actually just a layer of tissue that surrounds the lateral border of the kidney. After the renal capsule, the next layer is the perirenal fat. Then beyond the perirenal fat, we have the renal fascia, also known as the Gerota's fascia. And then finally, we have the pararenal fascia. So each of these structures are structures that tend to surround the kidneys at the lateral border, where the renal capsule, the renal fat, the renal fascia or Gerota's fascia, and then the pararenal fat or fascia. Let's talk about the internal structure of the kidney. So internally, the kidney has very unique features, and each of these features are shown here have names. They have particular content or structures found within them and particular roles. So what are the internal aspects or internal features of the kidney? So we have the renal cortex. All right, the renal cortex is this very light aspect of the kidney just beneath the renal capsule. So this very light aspect of the kidney is known as the renal cortex. Then we have the renal medulla. The renal medulla is this darker portion of the kidney that is found more centrally. So this darker area is the renal medulla. Then within the renal medulla, we have a combination of the renal pyramids and the renal colon. So the renal pyramids tend to be this inverted triangular structure found in the renal medulla. All right, so these are renal pyramids. You can see that we have a number of renal pyramids in this image. This is one, this is two, this is three. We have about 6 renal pyramids in this particular image. Between the renal pyramids is this extension of the renal cortex. So you can see the renal cortex, this whitish portion extends into the medulla, and separates the pyramids from one another. And this extension of the renal cortex into the medulla is known as the renal colon. So renal colon is this whitish aspect extending from the cortex, entering into the medulla and separating the pyramids from one another. Then at the tail end of the renal pyramids, we have this apex, the apex of the pyramids. Remember that pyramids tend to be inverted in shape. The apex of the pyramids tend to have this tail end that we call the renal papilla. So the renal papilla is the tail end of the renal pyramids, and they are all pointing to a central point of collection. So the central point of collection where all of the urine being produced at the renal papilla drains into is known as the renal calyx. So we can see the renal calyx is being the central point of collection. Each of the renal pyramids have their calyces. So we call those the calyces coming from the individual pyramids. We call them the minor calyces. And then when each of those minor calyces fuse together, they form a major calyx. The major calyx would eventually fuse and give rise to the renal pelvis. So the renal pelvis is this central point of collection. The renal pelvis automatically would drain into the ureter, which we'll talk about in the next slide. Let's talk about the renal cortex. The renal cortex is the outer part of the kidney in terms of its internal structure. It is a granular appearing structure, and so histologically it contains two major structures. It contains the renal corpuscle, and then it contains the convoluted tubule. So we'll talk about each of these when we get to the histology. The renal medulla is found in the inner aspect of the kidney. I said it is the hyperpigmented portion of the internal aspect of the kidney. It's composed of two parts: the renal pyramids, which I showed, and the renal colon, which is in between each of the pyramids. The renal medulla contains a combination of tubular and vascular structures. So their tubular and vascular structures are tend to arrange in a parallel radial line, and they tend to form the substance of the renal pyramids. So the renal pyramids, which are part of the renal medulla, pyramidal shaped dark medullary mass or structures. This contains a combination of tubular structures, particularly the loop of Henle and the collecting duct are found within the renal pyramids. We'll talk about each of these histological structures when we get to the aspect of histology in this model. The renal colon. The renal colon is also part of the renal medulla, the extension of the cortical tissue that tend to penetrate into the medulla. They lie between the two pyramids and they separate each of the pyramids from one another. Then we have the renal papilla. I mentioned this as being the apex of the renal pyramids. It serves as the final point where urine tends to actually collect at the collecting duct and eventually get excreted into the renal calyx. Then the renal calyces, like I said, these are the central collecting portion or collecting site for urine drainage within the kidney. They lie in that very, very unique space that I mentioned, found within the medial surface of the medial border of the kidney, the renal sinus. So they lie in the renal sinus. They're flattened tubes that tend to receive urine from the renal papilla and then eventually will drain into the renal pelvis. Remember, I said the renal calyces are divided into a minor calyx and a major calyx. The minor calyces tend to drain the individual pyramids, and then the major calyces are the combination of the minor calyces, and the major calyces will eventually drain into the renal pelvis. Let's end up talking about the renal pelvis. The renal pelvis tend to lie within the renal sinus, just like with calyces. This is a flattened funnel-shaped structure that receives urine from the major calyces and drains urine into the ureter. So just of note, it's important to note that within the renal sinus, we have a combination of the collecting system of the genitourinary tract, and this includes the renal pelvis, the renal calyx, and then some vascular structures like segmental vessels, the nerves, segmental nerves, segmental lymphatics, and then perineal fat. These are structures that are found within the renal sinus. Let's jump to the relations of the kidney. Remember I said the kidney has two major surfaces, anterior and posterior surface. Each of these surfaces tend to be related to structures surrounding the kidneys. Starting with the anterior surface. The anterior surface of the kidneys has a total of about 8 different structures related to it, and this differs in terms of the right and the left kidney. So for both kidneys, we have the suprarenal gland related to it, So you can see in both the right and the left kidney, we have a suprarenal gland found on the superior most aspect of the anterior surface of the right and left kidney. All right, now to distinguish them, for the right kidneys, the right kidney, we tend to see um, the duodenum, the liver, and so the duodenum and the liver are unique structures found or related to the right kidney. So you can see the hepatic area. This is the area that marks the location or the relation of the liver to the right kidney. And we can see the duodenal area. The duodenal area is the area that outlines where the duodenum is in relation to the right kidney. Besides the suprarenal gland, and other structures that are unique to both the right and left kidney would also include the small intestine and the colon. So the small intestine and the colon are quite uh, um, they are they are very related to both the right and left kidney. They are both related to the to both kidneys. So if you notice in this image, you see that the right and left kidneys have colic areas and jejunal areas. The colic area serves as the area for the colon, and then the jejunal area serves as the area for the small intestine. Then focusing on the the left kidney has organs or that are unique or exclusive to it. So this includes the gastric area for the stomach, the splenic area for the spleen, and then the pancreatic area for the pancreas. So these are structures that are unique to only the left kidney. Posteriorly, we have structures related to both the kidneys. This includes the diaphragm, the pleural membrane, the 11 to 12 ribs. Also, although for the left kidney, because it is higher up compared to the right kidney, we have the 11th and 12th rib related to the posterior surface of the left kidney. But for the right kidney, we have just the 12th rib related to the posterior surface of the right kidney. Then we have some muscular structures as well. So the muscular structures tend to be arranged in the vertical direction from lateral to medial. So from lateral to medial, we talk about the transversus abdominis muscle, then the quadratus lumborum muscle, then the psoas major muscle. So you can see each of them there, the transversus abdominis muscle, the quadratus lumborum muscle, and then finally the psoas muscles. Then finally we have some nerve structures related to the kidney posteriorly, so the subcostal nerve and the ilioinguinal nerve. Let's focus on the gross anatomy of the ureter. The ureter are also bilaterally placed retroperitoneal organs. So we have the right and the left ureter that are connected to the right and the left kidneys. And these structures are also lined anteriorly by peritoneum and not and lacks peritoneal lining posteriorly. The ureter tends to be measured about 25 centimeters in length, so extending from the renal pelvis higher up in the abdomen all the way down into the bladder in the pelvic region. Function of the ureter is in the function of transportation. It helps in transporting urine that has been produced at the kidney level to the urinary bladder for storage. In terms of location, the ureter tends to be located in two major parts. It's located in the abdomen, so we have an abdominal portion of the ureter. It's also located in the pelvis, we have a pelvic portion of the ureter. The part of the abdomen where the ureter is located tends to be the lumbar region of the abdomen, just like we have the kidneys being located in the lumbar region. The ureter begins in the lumbar region and crosses all the way down, enters through the iliac region, and eventually to the pelvis where it terminates. Let's talk about the extent of the ureter. Starting with the origin. It originates as a continuation of the renal pelvis. You can see the renal pelvis found within the ileum or the renal sinus, and the renal pelvis as a collection of the different calyces would um, combine together and eventually would drain into the ureter. You can see the ureter being a continuation of the renal pelvis. Once the ureter is formed at the pelvic region, it begins to descend. So it passes, first of all, along the medial part of the psoas major muscle on the posterior abdominal wall. Remember that the psoas major muscle is part of the posterior abdominal wall. So as the ureter is descending, it descends on the medial part of the psoas major and then runs from there through the abdomen into the pelvis. As it transits from the abdomen to the pelvis, it tends to cross the pelvic brim. Remember, the pelvic brim tends to distinguish between the pelvic cavity and the abdominal cavity. So it crosses the pelvic brim. At the pelvic brim, it tends to cross the common iliac bifurcation. So where we have the common iliac artery dividing into external and internal iliac, the ureter crosses anterior to the common iliac artery and also anterior to the sacroiliac joint. Then it enters into the pelvis as a result. Within the pelvis, it descends on the lateral wall of the pelvis and it descends very close to the iliac spine. Once it gets to the iliac spine, the ureter tends to be passed forward and then medially to enter into the bladder. There's a unique relationship between some structures of the pelvic region and ureter. We'll talk about that very soon. So this image depicts for us the course of the entire ureter. You can see it entering into the pelvis by passing through the pelvic brim, and you can see it crosses anterior to the common iliac artery at the pelvic brim. Then eventually it runs on the lateral wall of the pelvis, then runs towards the medial portion to drain into the bladder. While it is within the pelvis, we have this very unique relationship between the vas deferens in males or the uterine artery in females and the ureter. So we have this common mnemonic we tend to sing called water under the bridge. So water under the bridge practically describes the relationship between the ureter and the vas deferens or the uterine artery. So in females, the uterine artery runs above the ureter, and in this case, the water under the bridge in this case will mean the uterine artery is the bridge, and then the water tends to be the ureter. In males, it's the vas deferens that does the running. So the vas deferens is what is found above the ureter. So in this case, the vas deferens will be the bridge, the ureter will be the water. So finishing off with the extent of the ureter, it terminates by passing through the posterior wall of the bladder. It enters into the bladder, and the part of it that is found within the bladder is known as the intravesical portion of the ureter. It is approximately 2 centimeters in length, and its passage through the bladder is what produces a sphincteric-like effect, preventing backflow of urine from the bladder into the ureter and back into the kidneys. So it empties into the ureteric orifice and then transports urine into the bladder for storage. Moving on to the structures of the ureter. Structurally, we have the ureter being a muscular organ. It is made up of 3 parts and 3 constrictions. We'll talk about each of that very soon. So the 3 parts of the ureter are straightforward. We'll mention this already. It's just for us to recall them or review them again. So the 3 parts anatomically of the ureter includes the abdominal part, the part found within the abdomen. So that's the part spanning the lumbar region and iliac region of the abdomen. Then we have the pelvic part, the part that is found within the pelvic region, and then finally we have the intravesicular part, which is the part found within the has a short distance within the bladder. Then the constrictions of the ureter, there are 3 major constrictions of the ureter. These constrictions are formed at 3 main sites, and they include the constriction at the uretero-pelvic junction, the junction between the renal pelvis and the ureter. This is the first constriction. Then we have the second constriction, which is found at the level of the pelvic brim as the ureter transits from being an abdominal ureter to being a pelvic ureter. So that transition point forms this constriction. So in the pelvic brim, we have the second constriction. And then finally, we have the third constriction found as the ureter enters into the bladder. And that is known at the level, as found at the level of the vesical ureteric junction. So at this point, where the ureter enters into the bladder. So let's quickly talk blood supply. The kidneys tend to be supplied by the renal artery. The renal artery bilaterally placed structures coming from the abdominal ureter. We have the right renal artery and the left renal artery draining into or supplying blood into the kidneys. For the ureter, because we have the different parts of the ureter being abdominal and pelvic parts, these portions of the ureter will be supplied by different parts of blood vessels coming from different portions of the abdomen or of the pelvis. So the abdominal part of the ureter will be supplied by the aorta directly, the renal artery, and then the gonadal artery. So each of these arteries give the supply to the abdominal part. You can see vessels coming from the renal artery. You can see vessels coming directly from the abdominal aorta, we can see vessels coming directly from the gonadal artery, and this would also supply the abdominal part of the ureter. But when we get to the pelvis part of the ureter, this tends to be supplied also by separate arteries. So the pelvic part of the ureter is supplied by the internal iliac artery, by branches of the uterine artery, and then branches of the inferior vesical artery. So the branches of the internal iliac artery, you can see them um, emerging all the way lower down here, supplying the pelvic part of the ureter. And then depending on whether we're talking about a male or a female individual, if it's a female individual, the uterine artery will give supply to the ureter in the pelvic portion. If it's a male, the inferior vesical artery will give supply to the ureter in the pelvic part. Venous drainage is also quite straightforward. The renal vein tends to drain the kidneys, and it drains it into the inferior vena cava. For the ureter, it also has different veins that tend to drain it, depending on whether we're talking about the abdominal part or the pelvic part of the ureter. For the abdominal part of the ureter, we have the renal vein and the gonadal vein draining it. For the pelvic part of the ureter, we have the internal iliac vein, the uterine vein, and then the inferior vesical vein. Nerve supply. The kidneys and the ureter tend to supply by various nerve plexus. The kidneys would be supplied primarily by the renal nerve plexus. The ureter, on the other hand, is supplied by a combination of different plexi. This includes the renal nerve plexus, the abdominal aortic plexus, and the superior hypogastric plexus. And finally, lymphatics. Lymphatics also tend to be numerous, depending on whether we're talking about the kidneys or the different parts of the ureter. The kidneys are drained largely by the lumbar lymph nodes, whereas the ureter will be drained by a combination of the lumbar lymph nodes in the abdomen, the common iliac lymph node, and the external and internal iliac lymph nodes in the pelvis. So let's talk about the histology of the kidneys and the ureter. We'll start with the kidneys. So histologically, the kidney is composed of what we call a nephron. Each nephron is made up of two parts. So we have the renal corpuscle, which is found largely within the renal cortex. This is the renal corpuscle, and then finally, extending from the renal corpuscle, we have this long tail of tubular structure known as the renal tubule. It has numerous parts to it that we'll talk about next. So the first part of the nephron is the renal corpuscle. It's found within the renal cortex. It's a spherical blind ending part of the nephron that is involved in what we call the renal filtration of blood to form urine. There are two parts of the renal corpuscle. There's the glomerulus. Glomerulus is made up of a number of loops of capillaries that are derived from the renal artery. And then we have the Bowman's capsule. The Bowman's capsule is a epithelial part, is the epithelial portion of the renal corpuscle that eventually would extend to continue as the tubule. So this is the glomerulus found here in blue. It's lined by a number of a number of specialized type of cells called podocytes. And then this is the Bowman's capsule that you can see in the image here that eventually extends from the proximal convoluted tubule. The glomerulus, like I said, are types of capillaries that tend to arise from the renal artery. Particularly, they arise from a part of the division of renal artery known as the afferent arterioles, and eventually they will drain into the another form of arterial system known as an efferent arteriole. So they arise from the afferent arterioles and drain into the efferent arteriole. They're made up of a single layer of endothelium, and cells of the endothelium tend to be separated by spaces known as fenestrated pores. This allows for easy filtration of blood and the constituent of blood to form urine. Then we have the Bowman capsule. The Bowman's capsule is a cup-shaped capsular structure that tends to encase the entire glomerulus. So it's surrounding the glomerulus. It has two layers to it. There's a parietal layer and then there's a visceral layer. The parietal layer is made up of simple squamous epithelium and is continuous with the renal tubule. So it's a part that is continuous with the proximal convoluted tubule. The visceral layer is made up of what we call podocytes. That those blue structures I showed in the previous slide, and that's the part that tends to be in direct contact with the glomerulus, encasing it. as a result. Podocytes tend to be separated as well by small spaces, usually smaller than even what we see for the spaces between the endothelium of the glomerulus. This space is known as sleep pores. So the space found between the endothelium of the glomerulus are known as fenestrated pores, but the spaces found between the podocytes of the Bowman's capsule are known as sleep pores, and it also allows for the ultrafiltration that we see within the entire renal corpuscle. Let's talk about the renal tubules. The renal tubules are found both in the renal medulla and the cortex. So the portions of the renal tubules are found within the cortex, then portions extend into the renal medulla. The renal tubules are tubular in shape, hence the name. They are involved in the process of reabsorption and secretion of urine and its different in its various constituents. The renal tubules are composed of four major parts. So starting from proximal to distal, we have the proximal convoluted tubule. That's the part of the renal tubule in direct continuation with the glomerulus. Then, sorry, in direct continuation with the renal corpuscle, particularly the Bowman's capsule portion of the renal corpuscle. Then we have the loop of Henle. The loop of Henle is an extended part of the renal tubule that enters into the medulla, as you can see, and loops on itself. So you can see there's a descending part and then there's an ascending part of the loop of Henle. Then we have the distal convoluted tubule, which is just a mirror image of the proximal convoluted tubule, but found also in the cortex and also an extension of the loop of Henle. And then finally, we have the collecting duct that is extending from the cortex into the medulla and eventually draining into the renal papilla. We'll talk about each of these various aspects and then what is unique histologically to each of them. The proximal convoluted tubule, like I said, is the part or the most proximal part of the renal tubule. It's located in the renal cortex. It arises from the peripheral layer of the Bowman's capsule, so it's a direct continuation of that part. As you can see in this image, we have the glomerulus encased by the visceral part of the Bowman's capsule. This is the visceral part of the Bowman's capsule, and you can see the parietal part of the Bowman's capsule being a continuation or continuing as the proximal convoluted tubule. This is a tubule that is lined by specific epithelium, lined specially by simple cuboidal epithelium that contains unique modifications on its apical surface known as brush borders, and this is to allow for a greater range of absorption that we tend to see within the proximal convoluted tubule. Remember that the tubules are involved in renal reabsorption. So usually one of the greatest sites of renal reabsorption is at the proximal convoluted tubule, and this form of reabsorption is maximized by the presence of this apical modification we know as the brush borders. Let's go on to the loop of Henle. This is the second aspect of the renal tubule. It is a continuation of the proximal convoluted tubule. It is found within the renal medulla, and it eventually becomes the distal convoluted tubule. The loop of Henle has three major parts to it. It has a descending limb, then it has an hairpin bend, and then finally an ascending limb. The descending limb of the loop of Henle is lined by simple cuboidal epithelium with brush borders, so it's a direct combination or continuation from what we see in the proximal convoluted tubule. The ascending limb, on the other hand, is also lined by simple cuboidal epithelium but does not have a brush border, and this tends to mirror what we see in the distal convoluted tubule, as we'll talk about very soon. And finally, the hairpin bend between the descending and ascending limb is lined by simple squamous epithelium. The distal convoluted tubule is the third aspect of the renal tubule. It's located in the renal cortex, just like the proximal convoluted tubule. It arises from the loop of Henle and then eventually becomes the collecting duct. There are specific structures found within a distal convoluted tubule, not shown in this slide. These structures are known as the juxtaglomerular apparatus and the macula denta. These are important structures that tend to be involved in sensing various constituents within the urine and help in the regulation of blood pressure. That particular component of the distal convoluted tubule is unique, and it's important when talking about the concept of the RAS system, the renin angiotensin aldosterone system. That's a very unique system we'll talk about in physiology. But in anatomy, the actual structures are located within the distal convoluted tubule. This tubule is lined by a simple cuboid epithelium that does not have a brush border, so it mirrors what we see in the ascending loop of Henle. Finally, we have the collecting duct. This is the distalmost portion of the renal tubule. It's located in the renal medulla. It arises from the distal convoluted tubule, drains into the minor calyces of the renal papilla, and then it's lined by a simple cuboidal epithelium without brush borders as well. There are two major types of cells found in the collecting duct. We have the principal cells and the intercalated cells. Each of these cells are involved in various functions, some of which would be in the ability to sense um, the hormones aldosterone and antidiuretic hormone. These are hormones that are talked about in much more details in your physiology. Let's go on to talk about the histology of the ureter. The ureter is composed of a lumen and a wall that is surrounded or that is surrounding it. This wall is made up of three major layers from inside to outside. They include mucosa, the muscularis external layer, and then the adventitia layer. You can see an image of how the walls of the ureter tend to be formed and surround its lumen. The mucosa is the innermost layer of the ureter. It's composed of two major sublayers. This includes the epithelium and the lamina propria. And the epithelium of the ureter is unique because of its very distinct nature in comparison to other epithelium around the entire body. So the epithelium of the ureter tends to be lined by transitional epithelium, what we call uretilium. This is also found within the bladder and the urethra, and they are epitheliums that are unique to the entire urinary system. Then we have the muscularis external layer. The muscularis external layer is composed of three major sublayers of muscle. This includes the inner longitudinal muscle layer, the middle circular layer, and the outer longitudinal layer. It's important to note that the outer longitudinal muscle layer is absent in the upper two-thirds of the ureter. But when we jump down to the lower one-third of the ureter, we have all three layers present. The final layer of the ureter is the adventitia layer. It's the outermost layer composed of fibroelastic connective tissue and also contains a number of blood vessels, lymphatics, and nerves involved in the day-to-day action of the ureter. Let's end this section by talking about the embryology of the kidneys and the ureter. The kidneys and the ureter tend to develop from the intermediate mesoderm during the fourth week of the intrauterine life. Just to remind ourselves, during the fourth week, we have the bilaminar disc, the two-layered bilaminar disc becoming a three-layered trilaminar disc, and that process is known as gastrulation. So at the end of the or the start of the fourth week, we have the formation of three layers of tissue within the embryoblast. This includes the epiblast. This includes the ectoderm in blue, then the mesoderm in red, and then finally we have the endoderm in yellow. So these are the three layers of the trilaminar disc. The mesoderm gives rise to a number of structures. First of all, it's important to remember that the mesoderm gives rise, or first of all, divides into three major parts. The three parts of the mesoderm includes the paraxial mesoderm. This would form a number of skeletal muscle and skeletal and muscular structures of the body. Then we have the intermediate mesoderm found here in dark red. This gives rise to the entire genitourinary system. So it gives rise to the reproductive systems as well as the urinary system, which we're talking about now. And then finally, we have the lateral plate mesoderm, which gives rise to a number of body linings like the peritoneum, the pleural membrane, the pericardium, and otherwise. Our major focus is on the intermediate mesoderm because this is where the kidneys and the ureter would be derived from. So at about the fourth week of development, the intermediate mesoderm becomes the primordium of the urinary tract by forming a very important cord-like structure called the nephrogenic cord. So this cord tends to arise from the intermediate mesoderm and spans the entire length of the embryo. So it starts from the cranial or cervical region all the way down. So it starts from the cervical region specifically all the way down to the sacral region. And so this cord starts to develop into kidney sets, and it's important to note that at the various points in the embryo in embryology, we have different kidneys being formed. So there are three major sets of primitive kidneys that are formed during development before we eventually end up having our definitive kidney. The very first set of kidneys formed are known as the pronephros. Then we have the mesonephros formed later on, and then finally we settle at the metanephros, which eventually forms part of the adult kidney. So the pronephros arises around the fourth week, around the time in which the intermediate mesoderm begins to develop. So the very first set of kidneys that forms tend to be around the cervical region, so they're higher up, close to the neck. So at some point in time, we had the kidneys all the way up at the neck region. These kidneys were composed of three major parts. There's a pronephric glomeruli, just like we have our normal glomerulus. There was a pronephric tubule, just like we had our renal tubule, and there's a pronephric duct that connected them together. The better thing to note about the pronephros is the fact that it was a rudimentary or non-functional during its development. So, although it was present, it was not really doing any work in terms of its ability to function as a kidney. And about the ending of the fourth week, the pronephros tend to degenerate and is lost during the process of embryology. Then we have the mesonephros. The mesonephros also arises around the fourth week. It arises around the late fourth week. As the pronephros is degenerating, the mesonephros is eventually forming. It forms from the thoracic and lumbar region of the nephrogenic cord. So just as the pronephros is forming from the cervical or cranial part, the middle part of the nephrogenic cord gives rise to the mesonephros. Just like the pronephros, the mesonephros also has its own form of glomeruli, tubules, and ducts, and these are actually temporarily functional. So they function as a kidney during the development of the fetus in the embryo, in the uterus rather, up until a certain point in time. So up until a certain point in time, we have the mesonephros serving as the kidney. This is in contrast to what we saw for the pronephros, which doesn't have any function at all during development. So it's important to note that the glomerulus and the proximal part of the tubules and ducts of our mesonephros at around the 8th week begins to degenerate. So the mesonephros only works temporarily, and at around the 8th week it begins to degenerate. The part that tends to remain is the distalmost portion of the mesonephric tubules and duct. So the distal part of the mesonephric tubules, very close to the lumbar region, remains, whereas the rest of the mesonephros would degenerate. This distal portion of the mesonephric tubules and duct would eventually become structures that we'll talk about later on. Our major focus should be on the duct. So note the mesonephric duct, as it is terminating, it tends to empty into the hindgut in front of it. The hindgut is gotten from the end of them, and the portion of the hindgut where the mesonephric duct terminates at is known as the cloaca. Cloaca is an expanded portion of the hindgut. So please do note the mesonephros is gotten from the intermediate mesoderm, where most of the other urinary structures will be gotten from. The hindgut on this other hand, or the cloaca in this other hand, is gotten from the endoderm. So the endoderm is going to be connected to the mesoderm through the connection we see between the mesonephric duct and the cloaca. So the mesonephric duct empties into the cloaca. Just as it's about to enter into the cloaca, it gives off this outer growth or this out pouch. This out pouch or diverticulum is known as the ureteric bud. I'll talk about this very soon. It's going to be very important in the development of the definitive kidney. Final primitive kidney is the metanephros. The metanephros begins to develop around the 5th week. It is found lower down in the nephrogenic cord. It's found is located at the sacral region of the nephrogenic cord. It gives rise to the permanent kidney. It forms part of the permanent kidney. So it begins to function as at around the period in which it arises, but its functionality is permanent. So unlike the mesonephros, which is functional temporarily, the metanephros is functional continuously till we die. So until when the child is born, it continues to work until when the child eventually grows old and dies. That's when the metanephros ceases from functioning. Just like the metanephros also contains a metanephric glomeruli and a metanephric tubule. And the combination of both of these structure gives rise to a very unique structure we know as the metanephrogenic blastema. So when you hear metanephrogenic blastema, it just talks about the combination of the metanephric glomeruli and the metanephric tube. So the metanephrogenic blastema begins to interact with the ureteric bud. Remember I mentioned the ureteric bud being an outpouch extending from the mesonephric duct. So that bud runs towards the metanophrogenic blastema, and through the process of what we call reciprocal induction, both of them would mix together and give rise to the definitive kidney, the adult kidney. And we'll talk about the different aspect of the kidney that is gotten from the ureteric bud and the nephrogenic blastema. But what is important to note is that our adult kidney is a derivative that is formed from the combination of the ureteric bud and the metanephrogenic blastema. From this point henceforth, once the kidney is formed, it begins to ascend. Remember, the kidney is not found within the sacral region. Although our metanephros is found in the sacral region, the adult kidney is found within the lumbar region. So at some point, the kidney began to ascend from the sacral region to the lumbar region. And as it's ascending, it undergoes this form of rotation, what we call a 90 degrees medial rotation, whereby the the anterior part of the kidney becomes the medial portion and then eventually forms that medial surface or medial border that we mentioned at the start of the model. So this ascent is completed at around the 9th week of development. During the ascent, that 90 and then that 90 degrees rotation, the hilum, it was initially placed ventrally, but after ascent it becomes medially, and then as a result we tend to see most of the structures of the hilum found on the medial portion of the kidney. Essentially, with the derivatives of the kidney, the distal part of the mesonephric tubule will give rise to the efferent ductules of the testis in males. The distal portion of the mesonephric duct, that part that I said tends to remain, gives rise to what we call the seat structures. These are structures found in the male reproductive organ. They include the seminal vesicle, the epididymis, the ejaculatory duct, and then the ductus deferens. Usually, the features that we tend to see from these remnants of the distal part of the mesonephric tubules and duct in females tend to be remnants that are not really functional. So we don't really talk about them so much in terms of the anatomy. We'll learn them later on when we get to the module of reproduction. But in the case of this particular topic of kidneys and ureter, they're not really so important to note. Then focusing on the main derivatives, let's talk about the derivatives of the ureteric bud and the metanephrogenic blastema. For the ureteric bud, the part of the kidneys and the ureter that is gotten from the ureteric bud will include the collecting duct. Collecting duct would be derived from the first, or the proximal part derived from the ureteric bud. Then the minor connexus, major connexus, the renal pelvis, and the ureter. All of the structures are collecting structures that are gotten from the ureteric bud. The metanephrogenic blastema, whereas, would give rise to the adult structures of the kidneys, including the glomerulus of the kidney, the proximal convoluted tubule, the renal loop of Henle, and then the distal convoluted tubule. All of the structures are gotten from the metanephrogenic blastema. There's some important clinical significance to the topic of the kidneys and the ureter. We have renal agenesis, which is a lack of formation of the kidneys. Usually, children that have what we call a bilateral renal agenesis, where two of the kidneys are missing, don't tend to survive. They tend to die in utero or have stillbirth. A child, on the other hand, could have just one kidney, so we can have a unilateral renal agenesis, and that child could survive, but they always tend to come down with renal failure later on in life. Another form of clinical significance will be in the topic of horseshoe kidney or pancake kidney. So horseshoe kidney practically refers to a scenario where the lower poles of the kidney tend to be fused together. So instead of the kidneys being separate, a separate bilateral structure, they are usually one single structure fused together at their lower pole or inferior pole. We tend to see this in children that are that have been diagnosed with Turner syndrome. Turner syndrome, children tend to have high risk or high degree of horseshoe kidney. Another form of kidney is known as pancake kidney. This is when we have the superior poles fusing together and then forming one unilateral kidney. But we don't really see pancake kidney as often as we see horseshoe kidney. So horseshoe kidneys will be more common than pancake kidney. And just an important thing to remember is that the horseshoe kidney is related or associated with Turner syndrome. And that concludes our module on the kidney and the ureter.